21.2 Biotechnology. Remember, objectives are very important. After you're done studying, go back to the objectives to make sure that you covered the lesson properly. Again, biotechnology, as we just agreed in the previous lesson, is using microorganisms to produce required substances. Now we will take some examples of how does this work. Biotechnology, we can use anaerobic respiration by the microorganism yeast, or we can make enzymes by different microorganisms, mostly bacteria. If we use yeast, remember the anaerobic respiration by yeast used to produce ethanol and carbon dioxide. Ethanol, like in biofuel, we will use this ethanol to make a biofuel. We will see how in few seconds. And bread making, remember this from the anaerobic respiration chapter when we said that the carbon dioxide makes bubbles in the dough, and then this makes the baking spongy, right? What about making different enzymes? We need 3 different enzymes, or we will talk about 3 different enzymes. As we agreed before, enzymes are proteins. So taking the gene, placing it into the microorganism can make different proteins. In this examples, we are making different enzymes. Pectinase enzyme, protease enzyme, and lipase enzyme. Remember, the suffix ase means that it's an enzyme. We will use the pectinase enzyme in the juices industry, while we will use the protease and lipase enzymes in the detergents industry. Let's take a look about the details of each of these processes. This is a general map. Making mind maps is a very useful way to study lessons. So let's dig deeper into each part. Anaerobic respiration by yeast. Remember, we had different ways of anaerobic respiration, but we will focus here on yeast as a microorganism. So yeast can respire anaerobically, and we will have this equation. Glucose will be broken down into ethanol and carbon dioxide in the absence of oxygen. So ethanol for biofuels, this is the outcome of this equation that we will use now. How? So this is how we do it. First, from where will I get the glucose? I have 2 sources for the glucose, either the maize or the corn plant or sugarcane plant. Corn plants store their food in the form of starch. So in order to get glucose out of starch, we will use amylase enzyme. Remember the amylase enzyme from the digestive system chapter? So amylase enzyme usually works on starch to change starch into glucose. What about sugarcane? How does it store its own glucose? It stores it in the form of sucrose. Sucrose gets broken down into glucose. What is the enzyme? Without being mentioned in the slides, you can easily know it because you take the substrate and you add ase. So it is sucrase enzyme. So now we got glucose from those different plants. Then I will put my glucose and add yeast. Yeast will then do its anaerobic respiration, and it will produce ethanol and carbon dioxide. I'm not concerned about carbon dioxide now, but I need the ethanol. So I need to extract the ethanol from this mixture. What does this mixture have? It has carbon dioxide, yeast, glucose, and ethanol. So I will extract the ethanol by a process called distillation. So the process of distillation simply just takes out the ethanol from the mixture. Okay, so now I have ethanol. How will I produce a biofuel? I will mix it with petrol. I used petrol by the end of the day. Yes, but this will decrease the amount of petrol, which will enhance conservation. Remember, petrol was a non-renewable energy resource. So when we add ethanol, this will reduce the amount of petrol that we will use and will add a sustainable resource to it that we can produce quickly and replace quickly. What are the advantages of using a biofuel? First, the ethanol is a sustainable resource, so we can replace it quickly. We will always have yeast and we will always have glucose coming from plants, so this means that we can replace it quickly. When we grow maize and other plants a lot in order to get biofuel, this will increase photosynthesis because more plants, more photosynthesis. But there are some arguments against this. What are they? These are the crops that are taken for biofuel. These are using up a lot of land that people can use to grow more food. And also, this increases the monoculturing. Accordingly, monoculturing usually decreases biodiversity, which is not very good for the natural ecosystem. There is another argument is that when we use those crops for biofuels, and they should be originally food for humans, this means that this might increase their price as food. And this is another argument by naturalists. Okay, now we're done with the biofuel. Let's take the other side of the map, which is making bread. We already discussed this in the anaerobic respiration chapter, but let's go through it quickly again. First, grinding seeds of the cereal crop. What's the cereal crop? It's simply wheat to make flour. Then, this flour contains starch and gluten protein. We add yeast and water to the dough. And amylase enzyme will get released because we already grinded the seeds that contain the amylase enzyme naturally. So, some starch, by getting mixed with the amylase, will make maltose and glucose. And then glucose will be used by the yeast to make anaerobic respiration, which will release the bubbles of carbon dioxide that will make the dough spongy. And this is how we use yeast as a microorganism in order to make better baking or softer bread. Okay, now let's go to the other side of the whole map. Now let's see how do we use microorganisms to make enzymes. The first enzyme is pectinase enzyme. Since it's called pectinase enzyme, so the substrate is pectin. So what's pectin? It is a material that's found in the fruit fibers and the fruit cell walls. So when we add pectin to the fruit before squeezing it to turn it into juice, this breaks down its fibers and also breaks down its cell walls. So this, of course, will increase the amount of juice released. Instead of having some juice and some fibers, breaking down the fibers will increase the amount of juice because the whole fruit will be turned into juice. And since I have something that will break down the fruit fibers, I will get a clearer juice too, because no fibers will be left inside to make it turbid or to make it unclear. So this is how we use pectinase enzyme in the juice industry. Also, we use the biotechnology to make biological washing powders. We said that we here are making, yes, we are making enzymes. So we will make protease and lipase enzymes. Why? Because there are some stains that are made of proteins, like blood stains, for example. Remember, hemoglobin was a protein. So I need to break down this protein in order for the protein to leave the fiber. So this is how I wash it. It will turn into, the protein will turn into its monomer, and monomers are soluble in water. The monomer of protein is amino acid. So when I add the protease enzyme as a detergent to the water, it will break down the protein into amino acids. Now those small amino acids dissolve in water and leave the fiber. Lipase enzyme works the same way, but it breaks down fats or lipids into fatty acids and glycerol. So it works on the fats stains. Okay, you have to know something very important. Our skin has proteins and has lipids. So what happens if those detergents get in touch with our skin? Yes, this can cause serious injuries. That's why we pack those biological detergents into microorganisms capsules, so that when they touch the water, those microorganisms capsules will break down, and then they will do the work, or they will clean the clothes. But before touching the water, when you're handling them with your hands, they are sealed inside the capsules so that they do not damage the fingers. Okay, let's answer some questions together. Some washing powders contain enzymes as well as detergent. In an experiment, 3 pieces of cloth were stained with the same substance. They were left in different beakers of detergent, some with added enzyme, at 40 degrees Celsius for 30 minutes. Which statement is correct for the result shown in the experiment? So now, this one with detergent only, this one with detergent and lipase, and this one with detergent and protease. The stain disappeared in detergent and lipase. So this means that the stain was made of what? Yes, the black stain contained fats. Why? Because lipase is the enzyme that breaks down lipids or fats. Why is yeast used in bread making? Yes, to produce carbon dioxide that will make bubbles to make the baking spongy and soft. Yeast carries out anaerobic respiration, making carbon dioxide and ethanol as end products. Which end products can be used to make biofuel and bread? Yes, it's gonna be for biofuel, I need ethanol. For bread, I need carbon dioxide. A factory uses biotechnology to process apples. Some of the apple juice is mixed with yeast and used to make ethanol. State the type of respiration in yeast that produces ethanol. Yes, anaerobic respiration. State the name of the substance in apple juice that is converted into ethanol by yeast. Glucose. The yeast uses enzymes to produce ethanol. Define an enzyme. It is a protein that acts as a biological catalyst. Please read the question carefully, because reading the very first line of this question might make your head think of pectinase enzyme, because he's talking about apples, and so you may think about juice, or some apple juice is mixed, so and so. So please make sure that you read the question carefully to get the marks. Now, when you read the requirements, you can answer the question perfectly. Figure 6.1 shows apparatus used to find the best temperature making ethanol. The same apparatus was used at different temperatures. The rate of ethanol production was compared by counting the number of bubbles of gas produced by the yeast and apple juice mixture per minute. State the name of the gas produced by the yeast. Again, you might think that it's talking about the apple juice, so I will go to the pectinase enzyme, so and so. Please read the question carefully. Now, it is carbon dioxide gas produced by anaerobic respiration by the yeast. As a result, or the results of the investigation are shown in figure 6.2. Calculate the increase in the rate of reactions from 20 degrees Celsius to 25 degrees Celsius in figure 6.2. Space for working. This is a space provided for working. So first you will see from 20 to 25. This is 20 degrees Celsius. Here you go. It is almost 28 bubbles per minute. How did I know? Because here I have from 20 to 40. So this is 30. So each one of them has 2 bubbles per minute. So this is 22, 24, 26, 28. So it's almost 28 here. Okay? And 20 to 25. Where's 25? Some of you might think that it's here, but that's not correct, because we have 20 and 40. So in the middle, I have 30. So where's 25? It's in the middle here. So now I will go up and I will find out that it is 40 bubbles per minute. What will I do? I will subtract the 28 out of the 40, and I will get 12 bubbles per minute. And please do not forget to mention the unit if it wasn't written for you. Here the unit is written, but if the unit was not written, if you don't mention it in your answer, you will lose the whole mark. Please make sure that the unit is written. State the optimum temperature for the reaction shown in figure 6.2. Where do we see the optimum temperature? It is the temperature where the maximum rate of reaction is happening. So you will look at the top point of the curve, and it is the 60 bubbles per minute, and this is almost 35. So the answer is 35. Okay, the cut surface of an apple usually turns brown when exposed to air. This is caused by enzymes. 3 slices of apple were given different treatments and were then exposed to air for 30 minutes. Table 6.1 shows the treatments and appearance of the apple slices. If you look here, you will see treatment of pH or treatment of or changes of temperature and the outcome. Suggest why slice B and slice C did not turn brown after 30 minutes. So here let's look at slice B. What do you see? You see pH 2. So the treatment here was decreasing the pH dramatically to 2. This is an acidic pH, but the temperature was not changed. And the result was that the color did not change, which means that the enzyme was denatured. Let's take a look. Yes, because the pH was too low, therefore the enzymes were denatured. What about slice C? What was the treatment? It was the low temperature, but the pH stayed the same. Again, temperature is another factor that affects the reactivity of the enzyme. So, because the temperature was too low, therefore the enzymes were also denatured. The enzyme pectinase is used in the industry for the production of apple juice. Explain why pectinase is used when making apple juice. Now you will write the process of how pectinase enzyme works. So it is used to digest pectin, and it's better if you mention that pectin is a material that's found in, or a molecule that's found in the cell walls and fibers of plants, increasing the quantity of juice and also clarifying the juice. Thank you, and I hope this video helps you study.